start, right? So one can access the recording also. So the slides are visible.
Yes, sir.
Okay,
Yes,
okay,
sir.
okay. So we have already discussed about what is there in the syllabus, right, for the fifth semester, right? So we'll be basically starting with the antihistaminic agent today, right? So I'll be covering this first part today. What is the histamine? What are the receptor and how they are actually distributed in the body, right? Although I have discussed superficially in the first lecture, the introduction lecture, but today I'll be going into the details. So basically there are four types of histaminic receptor: H1, H2, H3 and H4. And in your syllabus you only have the two types of receptor, right? You have to only learn about the H1 antagonist and the H2 antagonist, right? So we'll be discussing about these things. So before starting, we are all familiar with the allergies, right? So we are all familiar with the allergies. So allergies are the one of the major thing where the main culprit is the histamine, right? So basically the question is like, do you have the spring allergies? Blame me, what molecule am I? Basically this is a riddle. So answer will be a histamine, right? So this is a histamine molecule. Let me open my pointer. Yes, this is the pointer. So see, basically, if you look at the structure of this thing, this molecule or the histamine, what we call, right? So this is a basically a histamine. So basically, this ring, what I can do here is I can just open the pen, right? For example, if you look at this ring, so you have learned about this ring in your fourth semester, right? So basically, it is an imidazole ring. Imidazole ring where basically it's a what 1,3 type of azole, right? So the example of 1,3 type of azole is imidazole. And here what we have three amine group. One, right? This is one group, this is second, and this is the third one, right? So basically it's a type of triamine molecule. Basically three amine groups are there, so basically it will be called as a triamine, right? So if you look at the scientifically, if you look at this thing, histamine is an organic triamine. It's an organic triamine, right? That is a strong vasodilator found in the blood and mostly bodily tissues, right? जो भी बॉडी के टिश्यूज है उस पे होता है. So basically इसका बेस रोल क्या है? Basically it is involved in inflammation, right? The H1 receptor are basically involved in the inflammatory response, right? और inflammatory response होगा क्यों? To combat the inflammation. For example, when there is any allergen, you are interacting with any allergen, so what will happen? There will be an inflammatory response. And why that inflammatory response is there, right? Because that is showcasing your immune response to that particular thing. Basically whenever body invade any foreign material, right? So there is an immune response against it, right? So immune response basically due to the histamine is by mediating the inflammatory pathways, right? So there is an inflammation when the histamine is involved in the immune response. So that is the important thing, right? So this we have discussed. Now if you look at the word histamine, there are two things: hist and amine, right? Amine we already know, it's an organic compound with the NS2, right? But what is this hist mean? Hist means the tissue, right? Hist means the tissue. For example, we are going for the histology, right? Histology is the study of the tissue. So basically the word histamine is derived from the two words, that is the tissue and the organic amine, right? So the word tissue means the histo, and the second thing is the amine, right? So the word histamine come into the picture, right? So this is the thing, right? So as I told earlier, ki H1 receptor is basically involved in the inflammatory response. The S2 receptor are the stimulant of the gastric secretion, right? So for example, if there is an ulcer, there is a cases of the ulcer, we require the S2 receptor antagonist, right? The third is the constrictor of bronchial smooth muscle, again the H1, vasodilator, again the H1, and centrally acting neurotransmitter. So when it is acting as a neurotransmitter, basically S3 and the H4 receptors comes into the picture, right? So basically S3 and H4 receptor are located in your CNS, right? Whereas I'll be discussing these all the things. So basically this is the common thing, right? So in your syllabus, basically there is H1 receptor and the S2 receptor, right? So I told you earlier that basically histamine is released against a immune response, right? Whenever there is a foreign material or something like that, so basically H1 receptor get activated and they release the histamine, right? And when there is a ulcer type of thing, the S2 receptor get activated and they release the HCl upon the release of histamine, right? So that is the thing. So now we comes to the biosynthesis of the histamine, how the histamine is synthesized in the body, right? So this is a very important question concerning your GPAT, right? The first thing, right? So basically the histamine is formed from the amino acid histidine, right? There is an amino acid called the histidine, and this histidine is responsible for the synthesis of histamine, right? So the enzyme involved here is histidine decarboxylase. See, if you look at the name decarboxylase mean to remove the carboxylic group, right? For example, see here it is a carboxylic group, COOH. I guess there was a lag now. There was a network issue, I guess.
Yes,
Yes,
sir.
sir.
Yeah. So I'll be just going to back, right? So this thing was clear. This thing was clear. Okay, okay. I'll be discussing about the metabolism, right? So I'll be again erasing all this thing, right? So basically what we have basically the we are talking about the biosynthesis, right? So biosynthesis whenever, so this is a very important question for the GPAT, right? So basically the histidine, the amino acid histidine is a precursor for the synthesis of the histamine, and this is done in the presence of a enzyme called the histidine decarboxylase, right? So I'll be opening that pen, right? Here it is. Right. So basically histidine decarboxylase is doing what? Decarboxylase. See, decarboxylase means removal of this carboxylic group. So if you are going to use this enzyme, so basically it will be removing this carboxylic group and you will be getting with this molecule. So basically this carboxylic acid is removed and what we have left with, we have left with the histamine. So basically this is the histamine, the imidazole with the three amine group, 1, 2 and 3. 1, 2 and the 3 amine group, right? But what is the problem with this histamine is that it has a very short life, right? So basically the inflammation or the immune response are very of very short duration, right? For example, a bee stings you, or if there is a pollen type allergy, right, sneezing or anything, so it is of very momentarily, it is a very small time, right? So basically this is because the histamine has a very short life, right? So this histamine is degraded by two methods, right? One is the oxidation and second is the methylation, right? So it is degraded rapidly, right? So that's why the histamine do not have a long T half, right? So this is the thing. So this is the thing. For example, histamine is there, right? So basically, histamine in the presence of, first I have talked about the methylation, right? First is the methylation, and the second is the oxidation. So both these processes are involved in the degradation of the histamine. So in the first thing, the histamine is there, right? So histamine in the presence of histamine and methyl transferase, right? So you can just correlate with the enzyme. See, and methyl transferase. So what is there? There is nitrogen, methyl transferase. So what is doing? It is transferring a methyl group to the nitrogen, right? So see, this is the histamine, and under the influence of this histamine and methyl transferase, the methyl is coming at this position, right? And why the methyl is coming at this position? Can anybody tell this thing? Why it is not going this way, this side, based upon your organic chemistry? Why this is not coming this side? Anyone? Why this is coming at this side only? The methyl is coming this side only. Anyone? Simple organic chemistry. Because there is this proton can be abstracted and can be replaced, right? So this is the very simple thing, right? Now the second thing, we talk about the oxidation. So basically in our body, right, there are basically two enzymes, monoamine oxidase and aldehyde dehydrogenase. So basically the monoamine oxidase and aldehyde dehydrogenase is responsible for the oxidation. So what will be happening in the oxidation? This ns2 group will be converted to the carboxylic acid group, right? So this is the fate of histamine in the body, right? So basically the key take messages from this thing is, basically it is synthesized from the amino acid histidine, right? This is the first thing, under the influence of histidine decarboxylase, right? The second thing, it get degraded rapidly, right? So it get degraded by what? It get degraded by two methods. One is the oxidation and the second is the methylation, right? So this is the important general thing you should remember, right? So now we'll be moving some little bit to the chemistry, right? Now again we'll be moving to your B Pharma fourth semester, right? So in the imidazole, you might be knowing about the tautomerism. You know about that thing, right? Tautomerism is there, right? So what is the tautomerism? Anyone? Anyone? On the basis of their knowledge of what we called B Pharm fourth semester, what is the tautomerism? Anyone? Anyone? Are you able to listen to me? So what is the tautomerism basically? Anyone?
So hydrogen is shared between nitrogens.
Means the shifting of the hydrogen, right? To basically it is an acidic hydrogen and basically it's a symmetrical type of molecule. So this hydrogen basically, what I'll be doing is I'll be opening the pen, right? So basically this proton or the hydrogen is transit between this nitrogen and this nitrogen, right? So basically this is the tautomerism. It can be the part of this nitrogen or it can be the part of this nitrogen. If I'm saying this as a position 1 and this is as a position 3, so the hydrogen can transit between the position number 1 and the position number 3. For example, in the second case, we have a hydrogen at the position number 3, right? If I look relatively from this position, right? So this is the tautomerism, right? So now if tautomerism is existing, right, the imidazole nitrogen, both the nitrogen can have a proton, means one at a time, right? So this is the tautomerism. Up now, how the tautomerism actually influences this thing? right? Influences the activity of histamine. So these are very important questions that may arise in your GPAT exam, right? So basic, they will not ask about the drug structure and anything like that, but they can ask about these concepts, right? So I'll be telling you one concept. For example, I have already discussed that. Let me erase this thing. Right. So I have already discussed the imidazole ring of histamine exists in two tautomeric form, identified by which of the two nitrogen atoms is protonated. Basically, which nitrogen is sharing the proton. For example, in this case, this one is sharing. In this case, this one is sharing. Right. So now there is, I have written something. I have written this pi sign. I have written this tau sign, and I have written the alpha sign. So now what is the significance of this thing? Right. So basically, when we looked at the structure of imidazole, right? So basically this hydrogen can be present at nitrogen 1 or it can be present at the nitrogen 3, right? Right? So basically the nitrogen farther away from the side chain is the tally nitrogen, right? This thing. This is the alpha. This is the alpha, right? So if this proton is at a some distance or it is away from this alpha amine, right? It is called the tally. It is called the tally nitrogen. This nitrogen is called the tally nitrogen. And one nitrogen which is close to this alpha, it is called the pi nitrogen, right? It is called the pi nitrogen, right? So what will happen now? See, the nitrogen farther away from the side chain is the tally nitrogen and is denoted by a lowercase tau sign. So this is the tau sign, right? This one is the tau sign. And the nitrogen closer to the side chain is the pros nitrogen and is denoted by the pi sign. So basically we are calling this as the pros and we are calling this as the tally, right? So it is denoted by a tau sign. It is denoted by a pi sign, right? So how we have to define this thing? The closer nitrogen with the alpha amine. The nitrogen which is close to the alpha amine is called the pros, right? And the nitrogen which is away from the alpha amine is called the tally nitrogen, right? So how this influence the activity, right? So we'll be discussing that thing. All right. So the tally tautomer and tau H histamine on the left, this one is more stable. This is more stable than the pros tautomer, right? Than this one, the second one, than the pros tautomer. So basically the N tally histamine is more stable, right? So now we'll be exploring the reason. Why is it so? Right. So basically the tally tautomer and tau histamine is preferred in the solution as compared to the prostautomer. Why it is preferred? Because it is more stable, right? So basically in aqueous solution, 80% of the histamine monocation exists as N tally H in the tally tautomer, right? And 20% as the pi tautomer. So can anybody give the reason why is it so? Why this is happening? Why basically this thing is happening? Why this is more stable? Can anybody tell in the solution form why this form is more stable based upon your chemistry knowledge? Anyone? What the solution will be doing? See, if there is a water or there is some buffer, what it will be doing? It will be abstracting the proton, right? It will be abstracting the proton. So why this moiety is more vulnerable as compared to this one? The tally is more stable and the pi or the pros is more unstable. So can anybody tell the reason for that thing? What is the reason? What could be the probable reason? Anyone? So what I'm going to do is I'm going to left this reason to you, right? So in the next class, what we are going to do, we are going to start with the reason first, right? So it will be an interactive, right? If you'll be discussing the reason first. So you will be telling me the reason. I'll be also explaining the reason. So you will be also, you have to find out the reason because there is a simple organic chemistry reason behind this thing, right? Why the tau histamine is more stable than the pros one, right? So you have to just search for this thing. right? Now in the pharmacology, have you read about the GPCR? Anyone? Have you studied about the G protein coupled receptor in the pharmacology? Anyone? Anyone knows about GPCR? What are the GPCR receptors?
Sir, they are membrane receptors.
Transmembrane, 7 transmembrane helical receptors. So you know about that thing, how they actually perform the mechanism. Something about that thing. So if you do not remember, see, basically, I think the GPCR was earlier in your syllabus, right? So what you have to do if you are not remembering the GPCR receptors, you have to just go back to the GPCR receptors, right? And just give a read about them, right? So basically, when we'll be dealing with the histamine receptors, when we will be dealing their mechanism of action, all the drugs will support or mediate via the GPCR mechanism, right? So this is the main thing. So basically, it will be very important that if you go back and just read about the GPCR receptors, right? So there is only one receptor that is involved in the histamine receptors, right? So if any questions comes in the GPAT exam, ki what is the basic receptor or what is the mechanism of these histamine receptors? So basically it is mediated by the GPCR, right? So this is the thing. So you have to just go back and just have to look at the GPCR receptor. What is that thing? How they are involved in the CAMP, how they are involved in the calcium release, right? You have to just check that thing, right? So if there is some problem, you can ask me anytime for that thing, right? So basically, we have 4 type of histamine receptor, as I told earlier also, right? So what is in your syllabus? In your syllabus, we have only these 2 receptors in the medicinal chemistry syllabus, right? So the important thing is you do not need to remember all this thing. What is the important thing is the H1 receptor. So where the H1 receptors are associated with, H1 receptor are associated with the allergic conditions and the sleep disorder, right? So this is one of the important thing, right? The S2 receptor are associated with the gastric acid secretion, right? And the S3 and the H4 receptor, for example, the S3 receptor are involved with the CNS disorder and the H4 receptor are involved with the major inflammatory pathways, right? The asthma and the rhinitis, right? So basically in your syllabus, you only have these 2 things, right? These 2 things, H1 receptor and the S2 receptor. So basically you should know if you are talking about the H1 receptors, right? If I'm talking about the H1 receptor antagonist, so basically they will be affecting your allergic conditions and the sleep disorder, and the S2 receptor antagonist will be selectively affecting your gastric acid secretion, right? So this is the common thing you must remember, right? Otherwise you will be giving all the wrong answer in your exams and other things, right? So this is a very common thing. H1 are associated with the allergic conditions and the S2 are associated with the gastric acid secretion, right? So this is the thing, right? Now if you might be remembering the GPCR receptors, right? You might be remembering the mechanism of GPCR. So basically the H1 receptors coupled to the GQ, right? There are the subtypes of the GPCR receptors. So GQ. So basically GQ is involved in the regulation of Ca2, calcium 2 mobilization. So basically if H1 receptor, what is their mechanism? They actually couples to the GQ receptor. GQ receptor is the subtype of the GPCR, right? So they are involved in the calcium mobilization, right? So if we are taking the H1 antagonist, what will happen? Basically, this calcium mobilization will stop and the histamine will not get released, right? So this is the first mechanism. The second thing, the H2 receptor, this one, the H2 receptor coupled to the GS stimulatory GPCR, right, to stimulate cyclic AMP, right? So this is the thing. So you have to just remember this small thing. The H1 receptor will affect the calcium mobilization, means calcium se kya hoga? They will be bursting the mast cell and the WBC cell to release the histamine, right? The second thing, the histamine 2 receptor will be coupled to the GS and they will be stimulating the cyclic AMP, which will be then regulating the release of HCl in the stomach, right? So these two things are there, right? Then S3 and H4 you can go if you want to read it, right? So basically there are the two receptor H1 and S2 in your syllabus. So I'll be just discussing majorly about these things, right? So now just summary of what we have done today, right? So basically what we have the four receptor H1, S2, S3 and H4, right? So this is a self-explanatory figure, right? So basically all the allergic and the inflammation, right? It's covered by the H1 receptor, right? All the gastric acid secretion is covered by the S2 receptor, right? So if we want to target the allergic inflammation, what we have to do? We have to block the H1 receptors. If we want to block the HCl release in the stomach, what we have to do? We have to block the H2 receptor, right? So this is the simple thing. This is the simple thing, right? So this was the general thing. So basically the H2 receptor will regulate the gastric acid secretion and the H1 receptor will improve the vascular permeability, right? So this is the thing. So now what we have the H1 antagonist. So H1 antagonist, I'll be starting from tomorrow, right? So I'll be just telling you about the basic structure and we'll be doing some classification and the basics are, right? What are the important function group there to for the activity and other thing, right? So in the meantime, if you have any questions regarding to what we have learned today, you may ask. Any questions, queries, doubts? If you are not understanding something, so I'll be explaining that thing. Anyone? Any questions, doubts, anything? If you
No,
have,
sir.
other than hers, if someone is having doubt. Sorry, I just pronounced your name wrong. So any doubts, queries, anyone? Were you able to understand the concept of that tally and the pros? समझ में आया वो चीज समझ में आया, that is the important thing, right? So the question you can expect from this chapter in the GPAT are basically the amino acid which is involved in the synthesis of that histamine. That is the first question. The second question that can be in your GPAT exam, which form is more stable of the histamine, whether it is the pros form or whether it is the tele form, right? So the reason you have to tell me on the next class. I'll be also sharing what are the actual reason for that thing, and then we'll be moving to the H1 antagonist. The third question that may come in the exam is from the degradation, right? So basically the two methods are involved in the degradation. One is the methylation and the second is the oxidation. So in the methylation, what is happening? A methyl group is attaching at the NH position, right? The hydrogen is replaced by the methyl. And in the carboxylation, what is there? Basically the NH2 group is converted to the carboxylic acid, right? So this is the simple mechanism, right? So if anyone have any questions, queries or doubts, they may ask. So I'll be uploading the books and the resources in your Moodle soon. Huh? So currently there is some issues with the Moodle. So as soon as they get over, so I'll be putting something, the books and these notes and other thing, right? So you can go through them and you can ask the question. So from tomorrow we'll be starting the chemistry of this H1 antagonist, right? So this is the way how I will approach basically for your medicinal chemistry. If you want me to change something, so I can do because this is the early time, right? So otherwise you will be learning these only things. So I think it was very basic. It was very basic and you were able to grasp the concept what I was going to tell you, what I was telling you actually. So that was the thing. Yeah, I need to change something in that thing. Anyone can give their frank opinion.
So this method is fine.
This method is fine. Right. Right. So from tomorrow, what we are going to start, we are going to start with the H1 antagonists, right? So there are many H1 antagonists in your syllabus. So we'll be discussing, and we'll be not discussing like drug by drug. I'll be doing what? I'll be just categorizing them in some medicinal chemistry category. So chemically, I will be just classifying them, right? In the pharmacology, we basically classify the histamine receptor as like this. Either we talk like the H1 antagonist, H2 antagonist, or the S3 antagonist, or first generation H1 inhibitor, second generation H1 inhibitor. But in medicinal chemistry, we are not going to approach like that, right? We'll be actually defining the classification on the basis of the pharmacophore, right? So that is the thing we are going to do, right? So then if there is no queries or nothing, so I'll be just signing off this class, right? Thank you for joining.
Thank you, sir.
Thank you, sir.
Thank you,
Thank you,
sir.
sir.
Thank you, sir.
If you have any doubts or queries, you may ask.